Embryology of the head, face and oral cavity. At this point, the rostral or head fold is important. The anterior portion of the neural tube expands greatly as the forebrain, midbrain and hindbrain form. The part of the neural tube that forms the hindbrain develops a series of 8 bulges referred to as rhombomeres. Migration of neural crest cells provides the embryonic connective tissue needed for craniofacial development. These neural crest cells arise from the midbrain and the first 2 rhombomeres as 2 streams. The first stream migrates forward and intermingles with and reinforces the mesenchyme situated beneath the expanding forebrain. This stream provides much of the connective tissue associated with the face. The second stream is directed towards the first branchial arch. These migrating streams of neural crest cells continue to express the homeobox genes that were expressed in the rhombomeres from which they were derived. Development of branchial or pharyngeal arches and the primitive mouth. When the stomatodeum first forms, it is limited by the frontal prominence rostrally and caudally by the developing cardiac bulge. The buccopharyngeal membrane, which is a bilaminar structure, consists of apposed ectoderm and endoderm. This buccopharyngeal membrane separates the stomatodeum from the foregut. This soon breaks down so that the stomatodeum communicates directly with the foregut. Laterally, the stomatodeum is limited by the first pair of pharyngeal or branchial arches. As a result of proliferating lateral place mesoderm and subsequent reinforcement by migrating neural crest cells, branchial arches form in the pharyngeal wall and result in 6 cylindrical thickenings. Note that arches 5 and 6 are transient structures in humans and are not visible on the surface of the embryo. These thickenings expand from the lateral wall of the pharynx, pass beneath the floor of the pharynx and approach their anatomic counterpart, which is expanding from the opposite side. Hence the arches progressively separate the primitive stomatodeum from the developing heart. The arches are clearly seen as bulges on the lateral aspect of the embryo and are separated externally by small clefts called branchial grooves. On the inner aspect of the pharyngeal wall, we see corresponding small depressions called pharyngeal pouches that separate each of the branchial arches internally. Each branchial arch has the same basic plan. The inner aspect is covered by endoderm, but in the case of the first arch, the inner lining is ectoderm because it forms in front of the buccopharyngeal membrane. The outer surface is covered by ectoderm. The central core consists of mesenchyme derived from lateral plate mesoderm. This area is invaded by mesoderm from the neural crest referred to as ectomesenchyme. This ectomesenchyme condenses to form a bar of cartilage referred to as the arch cartilage. The cartilage of the first arch is called Meckel's cartilage, and that of the second arch is called Reichert's cartilage. The mesenchyme surrounding the first and second arch cartilages give rise to the muscles of mastication and to the muscles of facial expression respectively. each arch also contains an artery and a nerve. The first, second, and third branchial arches play an important role in the development of the face, mouth, and tongue. The first, second, and third branchial arches play an important role in the development of the face, mouth, and tongue. The formation of the face is described in terms of the formation and merging of several processes and prominences. The term merging is described as swelling of processes with the elimination of a furrow between them. Fusion, on the other hand, occurs in the union of the palatal processes. Such fusion involves the breakdown of surface epithelium. The primitive stomodium is at first bound by the frontal process rostrally, caudally by the developing heart, and laterally by the first branchial arch. With spread of the arches mid-ventrally, the cardiac plate is eliminated from the stomodium, and the floor of the mouth is formed by the epithelium covering the mesenchyme of the first, second, and third branchial arches. At about 24 days, the first branchial arch establishes another process, the maxillary process. Now the stomodium is limited cranially by the frontal prominence, which covers the rapidly expanding forebrain, laterally by the newly formed maxillary process, and ventrally by the first arch, which now becomes known as the mandibular process. Early development of the face is dominated by the proliferation and migration of ectomesenchyme involved in the formation of the primitive nasal cavities. At about 28 days, localized thickenings develop within the ectoderm of the frontal process, just rostral to the opening of the stomodium. These thickenings are referred to as olfactory placodes. Rapid proliferation of the underlying mesenchyme around these olfactory placodes results in bulges of the frontal eminence and also produces a horseshoe-shaped ridge that converts the olfactory placodes into nasal pits. The lateral aspect of the horseshoe is called the lateral nasal process, and the medial aspect, the medial nasal process. The region of the frontal prominence where the nose will develop is referred to as the frontonasal process. The medial nasal processes of both sides, together with the frontonasal process, gives rise to the middle portion of the nose, the middle portion of the upper lip, and the anterior portion of the maxilla, as well as the primary palate. The maxillary process grows medially and approaches the lateral and medial nasal processes, but remains separated from them by two distinct grooves, the naso-optic groove and the bucco-nasal groove. The medial growth of the maxillary process pushes the medial nasal process towards the midline, where it then merges with its anatomic counterpart from the opposite side. The upper lip is therefore formed from the maxillary process of each side and the medial nasal process. The lower lip is formed by merging of the two streams of ectomesenchyme of the mandibular process. Note The merging of the two medial nasal processes results in the formation of that part of the maxilla that carries the incisor teeth and the primary palate, and also a part of the lip. At about 30 days of gestation, the epithelium on the inferior border of the maxillary process and the superior border of the mandibular arch begins to proliferate and thicken. This thickened area is referred to as the odontogenic epithelium. At this time, the primary epithelial band can also be observed. The primary epithelial band is a single plate of thickened epithelium described as an arch-shaped continuous plate of odontogenic epithelium. In the upper jaw, it forms from 4 separate zones of epithelial proliferation. That is, laterally, one zone associated with each maxillary process, and in the middle one with each medial nasal process. In the lower jaw, it forms from 2 zones, that is, one from each mandibular process. At this time, we have a common oronasal cavity. This is bound anteriorly by the primary palate, and the cavity is occupied mainly by the developing tongue. Only after the development of the secondary palate does one get the distinction between the oral and the nasal cavities. The palate proper develops from the primary and secondary components. The formation of the secondary palate commences between 7 and 8 weeks and completes around the 3rd month of gestation. 3 outgrowths appear in the oral cavity. The nasal septum grows downward from the frontonasal process along the midline, and 2 palatine shelves, one from each side, extend from the maxillary processes towards the midline. At first, the shelves are directed downward on each side of the tongue. After the 7th week, the head of the embryo tilts back, the tongue is withdrawn from between the shelves. These shelves then elevate and fuse with each other above the tongue and anteriorly with the primary palate. The septum and the 2 shelves converge, fuse along the midline, and hence separate the primary oronasal cavity into nasal and oral cavities. The tongue begins to develop at about 4 weeks. The pharyngeal arches meet in the midline beneath the primitive mouth. Local proliferation of ectomesenchyme then gives rise to a number of swellings in the floor of the mouth. The first swelling arises in the midline of the mandibular process. This is referred to as the tuberculum impar. This is then flanked by 2 other bulges referred to as the lingual swellings. These lateral swellings enlarge and merge with each other and with the tuberculum impar to form a large mass from which the mucous membrane of the anterior 2/3 of the tongue is formed. The posterior third of the tongue arises from a large midline swelling developed from mesenchyme of the 2nd, 3rd, and 4th arches. This swelling consists of the copula, which is associated with the 2nd arch, and a large hypobranchial eminence associated with the 3rd and 4th arches. As the tongue develops, the hypobranchial eminence overgrows the copula, which then subsequently disappears. The posterior part of the 4th arch marks the development of the epiglottis. The tongue separates from the floor of the mouth by a downgrowth of ectoderm around its periphery, which then subsequently degenerates to form the lingual sulcus. This allows mobility of the tongue. The muscles of the tongue have a different origin. They arise from the occipital somites, which have migrated forward into the tongue area, carrying with them their nerve supply, the 12th cranial nerve or the hypobranchial nerve. Development of the mandible. In humans, Meckel's cartilage has a close positional relationship to the developing mandible, but it makes no contribution to it. At 6 weeks of development, this cartilage extends as a solid hyaline cartilaginous rod surrounded by a fibrocellular cartilage. It extends from the developing ear region to the midline of the merged mandibular processes. The cartilage from each side does not meet at the midline. They are separated by a thin band of ectomesenchyme. The mandibular branch of the trigeminal nerve, which is the nerve of the first arch, has a close relationship to Meckel's cartilage. It begins 2/3 of the way along the length of the cartilage. At this point, the mandibular nerve divides into the lingual and the inferior alveolar branches, which run along the medial and lateral aspects of the cartilage respectively. The inferior alveolar nerve further divides into the incisor and mental branches anteriorly. During the 6th week of embryonic development, on the lateral aspect of Meckel's cartilage, a condensation of ectomesenchyme occurs in the angle formed by the division of the inferior alveolar and its incisal and mental branches. At 7 weeks, intramembranous ossification begins in this area of condensation, hence forming the first bone of the mandible via intramembranous ossification. By 10 weeks, the rudimentary mandible is formed almost entirely by intramembranous ossification with little or no direct involvement of Meckel's cartilage. Further growth of the mandible until birth is strongly influenced by the appearance of 3 secondary or growth cartilages and the development of muscular attachments. Development of the maxilla. The maxilla also develops from a center of ossification in the ectomesenchyme of the maxillary process of the first arch. No arch cartilage or primary cartilage exists in the maxillary process, but the center of ossification appears in the angle between the divisions of a nerve, where the anterosuperior dental nerve is given off from the inferior orbital nerve. A secondary cartilage called the zygomatic or malar cartilage, which develops in the zygomatic process, also contributes to the development of the maxilla. At birth, the body of the maxilla is relatively small because the maxillary sinus has not fully developed. The maxillary sinus forms during the 16th week of development as a shallow groove on the nasal aspect of the developing maxilla. At birth, the sinus is still a rudimentary structure about the size of a small pea.